Hi, in this video I am going to demonstrate how we can isolate samples based upon a lack of a mutation, which is always a challenging topic, and how we can use that as part of designing differential expression analyses that use data from the genomic data commons, specifically using data from the TCGA. Now, not to just date this video, but I do want to draw your attention that this is an active research tool, and as such, it does occasionally go down for maintenance. Now, unfortunately, this is the week that some of my project stuff is due, so there'll be more about that later, students, this semester. But whenever this happens, you do have to pay attention. And so I'm going to dismiss this, but it's always important to check up here when you log in. Okay, so there's a few setup things just to start off with. I'm going to point out that my cart is empty. That's good. That's actually important. I'm going to remove some saved data from here just so it looks a little bit more like your data. So starting off, you should have something that's pretty clean. So you don't need to do that. I was just making sure that it's understandable to everyone. Now, this is the main view. I have some other videos for actually just exploring the GDC in general, so I'm going to jump right into the goal of my data. So the goal of my experiment is that I want to look at colorectal cancer. And so I'm going to open up the COAD from the TCGA. Whenever I do that, it updates everything around here. So it's like the project ID is now TGCA or TCGA, which means the only program that's available is TCGA. There are a lot of important filters that tend to occur in this cases. So for example, the primary site is going to be very important. Like if you're in a different tissue, well, yeah, different tissues have different gene expression profiles. And so you need to be specific in regards to primary site. It's kind of a similar situation for disease. That's a major way that we categorize what's going on with these people and their pathologies. And so that's going to be very commonly a super important control that you want to be the same across all of your samples. So here I'm picking adenomas and adenocarcinomas. Finally, most people in my class are going to be interested in studying primary tumors, as that's one of the major data sets that's available. Example here, though, there is a blood-derived normal sample. That's actually probably a bad grouping to use, and that's because, like, what are you going to compare it to? Now, you can compare normal blood from someone with a low severity to normal blood from someone with a high severity. That'd be a great project, but you don't really want to compare blood to tumor, because the answer is going to be a lot, and it's going to be mostly to do with the difference between these types of the tissues. That it's blood, it's got different genetics than tumors do. Kind of similar to metastatic and recurrent tumors, there's not really options here. Solid tissue normal is actually a pretty great comparison against primary tumors. It's usually very interesting, especially for the earlier stage cancers. Late stage cancer, it's kind of like, let me compare a car accident to a healthy person. The answer is a lot of differences, but it's not necessarily informative of everything going on. So anyways, I'm going to select primary tumor. Down here we have SSM and CNV. CNV's copy number variant. You can work with that data, but that's not necessary. It's harder to use. Simple somatic mutations are understandable. You've learned about them in your genetics classes. So that's what I recommend most students who are interested in investigating specific genes, that's what they follow. So I'm going to pick that one. Okay. So at this point, I have cases, I have all my controls that I want. I could probably go to clinical, and because I am working with colorectal cancer, which is known to be highly gender influenced, I'm going to probably select male samples just to help control for that. Finally, I might be interested in controlling for pathologic stage. Like I said earlier, you often want to control for pathologic stage because as you progress, a lot of stuff starts to change. So it's just something that always bears consideration. So I might pick like all stage 2 or all stage 3 or a portion of stage 3. So I think C kind of starts to get a little bit wild itself. But if you're interested in the differences, they are often kind of cancer specific, although there are certain patterns. So like from 3 to 4 is the transition to multiple metastatic sites. That's what makes it 4A, is multiple metastatic sites have been found. So I mean, it's a definite like transitionary period in terms of the cancer going from one stage to another. Within a stage, there are more subtle differences, and you can see that there's a good number of samples not broken down into individual stages. So just things to bear in mind. So I'm going to leave these as all of my kind of controlled cases. So any of these cases I find to be somewhat comparable. You know what, let's just see what happens if I make it just stage 2. I'm probably going to regret it. This is a video and you shouldn't be doing live analyses. Actually, let's make it stage 3. Oh no, stage 2 is fine. Okay, so stage 2, stage 2, stage 2, stage 2C. Yeah, let's keep it in. Okay, so we're going to call this the like default setting. So these are everything that I think is mostly comparable. Let's see how we can break them down. So I'm going to say, I'm just actually going to leave this, but I'm going to add control set to the beginning. Maybe total control set. So everything from the rest of this, I want to be part of these 72. So there's a lot of sections to this website. The main thing I've been interested in studying is what are the differences between samples that lack mutations in APC and samples that have mutations in APC, because APC is tremendously associated with colorectal cancer. You really need it to be mutated in the vast majority of cases in order for the cancer to have progressed. That's why it's somewhat important for me to have selected for the stage, because I need samples that have progressed through the disease, because that's when APC really matters. So if I pick that, I have 72 minus 50. That's how many samples lack simple somatic mutations. And so 50 is not going to be my comparison, but I will need that number in order to find the absence of mutations in APCs, because that will be one of my groupings. So I should have a 22 member group, or approximately 22, depending upon how many of those people had genetic expression data collected. So to do that, I've already selected the 72 individuals as a case set. So I'm going to click the 50. I'm going to save this 50 as APC mutants. I'm going to say all APC mutants, because we're going to have a third set that is just specific people within this 50. So within this, I am going to just go to those people that have high impact mutations in any of the genes here. And since I have already filtered this down to just the APC gene, when I filter down the list of mutations to just high impact mutations, I'm left with the 49 individuals from that original, I don't remember what it was, 70? I don't remember, whatever it was originally. I have the 49 individuals from that subset of all individuals with mutations that have at least one high impact mutation. And honestly, they often have other mutations as well. But I want the high impact because that's going to be mutations, things like frame shifts that are very likely to prevent gene functioning. And that's probably why they're now getting cancer, is APC being mutated alongside genes like TP53 and other genes kind of necessary for tumor production. That's what's going on. Okay, so I am going to save this. This is actually going to be one of my experimental sets. So this will be APC high impact mutant. I just kind of keep some of the rest of that in there just to help me recreate things if needed. And finally, I need to go back and I need to get that subtractive kind of set of data, those samples that had no mutations in that gene. They have plenty of mutations. They're still tumor samples, but they just don't have mutations in that gene. So let's say there was only one sample that had some mutations, but not a high impact mutation, at least within the set of data that I had selected. So what I'm going to do is, let me just select all three. I think that's helpful. I think maybe it confused people this semester, but like there's one sample in that 50 that was all probably like moderate impact mutations. But just for clarity, I like to separate anything that doesn't at least have one high impact. It's not necessarily always true, so I know that these are people who have these diseases. I know that APC is a really big part of this, and the fact that they have an APC mutation at all probably implies that it's a fairly impactful APC mutation. It's probably why they got this cancer, or at least correlated with the fact that they have this cancer. That said, it still makes my analysis a little bit more ambiguous because they could still be partially functional, whereas these 49 are at least suspected that one of their two copies, at least, has a non-functional copy of APC. So these 49 are going to be one of my groupings, and then out of all of those, 22, remember I said that at the beginning, that's 72 minus the 50 that had any mutation, 22 lack any mutation at all. And so that will be my final set. So this would be, I don't know, like TCA, GA, co-ed, controls without APC mutations. So they lack mutations in APC. Okay. So at this point, I have like the cases I'm interested in. I've saved them for easy reference later on. So here's my 22, here's my high impact. What I now need to do is download these into a sample set so that I can extract their identifying information for use in R. So here I've just clicked on that number 22 that pulls up the exploration tool and allows me to then move into the repository to see the files associated with these cases. And there's going to be a lot of files. So 1,228. Not quite 10 times as much, but it's a lot of data. So for each of these different cases, I said 10, like not, I was off by an order of magnitude, not 100 times, but almost. Anyways, so structural variation, not what I want. Transcriptome profiling, transcriptome referring to the transcripts produced in the genome. Yes, what we're doing, a gene expression analysis, is a form of transcriptome profiling, at least according to their definition set. Within that, there's multiple types of data that can be collected. Some of these are very interesting. Some of these I know how to work with. Some of them I don't. But gene expression is the type of data we're looking for differential gene expression analysis. So I could keep clicking. The rest of these don't really matter because I can already see there's only one for each of these, and they're all 26. But a very important one is this open access. Again, there wasn't any closed access, but it's always one to be aware of. In the end, just for the record, what you're downloading is star count data. It's a program used to count how many reads belong to a gene. So it's kind of, there's some ambiguities there. There's different ways to summarize how reads are aligned to genes. So here I can see there's 25 files associated with these 22 cases. What that tells me is that some of the cases have additional files. It could be things like metastatic tumors, recurrent tumors. Because I've only identified these things based on the cases, later on I'm going to have to make sure I remove any like non-primary tumors that I care about. And that's just a limitation of the GDC. There isn't a button to allow me to, at this point, when getting the file information, isolate just those samples that I want. So what I'm going to do is I'm going to take all of this without having kind of selected any of this. I'm going to hit this Add All Files to Cart button. When I do that, I can now go to my cart. That's why it was important that my cart was empty. And I'm going to click Sample Sheet, not Download. Downloading these directly is very difficult, and it can cause a lot of problems. I mean, it's always going to be hard. That's just one of the facts of this. And so, just for the record, this was the method I have found to be the best. I may eventually find a better way, but this is one of the better ways to do this that I have found, at least that uses R primarily. So you're going to download the sample sheet. Let's see. Get out of full screen mode. And there is my current sample sheet. I almost opened the wrong one, got real close. I'm going to open it in Excel. You can open it in a lot of other things as well. So I can see here I have 1, 2, 3 solid tissue normals. I'm probably just going to delete those in here. Let me make that a little bit bigger. Just selecting out on the number so that it selects the whole row. And I've just deleted that. And that's left me with 22 samples. I could check a few other things. I could make sure that there aren't any duplicates. So cool, there aren't any duplicates. I could check my sample IDs. Again, just make sure there's no duplicates. Good, still no duplicates. These are just kind of common issues that could arise later. That all said, it looks good. I've got the expected number of samples. And so I am going to save that, and then this would become the input for one of my grouping objects in R. So this is what I would use: read.delim, maybe read.table. Read.delim is better for these files, is what I found. I think they used to use an underscore here, but now it's a space. And so if you use read.delim, this becomes a single column, as it should be. But anyways, so that's what I will do with this data in order to get the second grouping. Oh my gosh. In order to get the second grouping, I would clear this out, and I will note that don't use multiple tabs. It does not work well. It causes challenges that you don't really want to work with. So I've removed those. My cart is empty. I'm going to double click it just to make sure. And I'm going to go back, do that same process, but much faster. Transcriptome profiling, gene expression, check the open, they're all open, cool. Send it to my cart, go to my cart, download the sample data. Clean up the sample data. I can see that there's a number of primary of solid tissue normals in here. If this was going to take too long, I could sort this. I'll show you sorting in a second. This is technically very close to right. I would be fine, but let's pretend this is more than just 51. So 50 is what I recommend as the maximum size. If we pretend this was like, here, I'm just going to artificially do it. Oh no, my file is so large, it's not fake at all. If it was 102 samples large, that would be kind of stressful to the system. And so what I could do is I could use Excel to randomly select some. So I'm going to just run the rand function. Oh my gosh, get bigger. There we go. So the rand function, start a function in Excel with an equal sign, then the function is just rand, and then it's open parentheses, close parentheses, and it generates a random number. I'm going to put my cursor right here until it turns into this little cross shape, and then double click it, carrying it all the way down. You notice that every time I kind of manipulate this, all the numbers update. So it's just like a way to randomly generate data. And then if I go into sort, so I've selected everything, including the header. I then selected my data has header. and I'm going to sort by that column of random values. When I do that, I've now randomly selected things, and I can just choose the top 50 or so that I want. So there we go. There is one problem with this, is that sometimes when you save it at this point, if you were in a program like Excel or Google Sheets or Numbers, you'll have added a bunch of empty cells to the bottom. And so just always, my recommendation when exporting to a simple file type, actually, is that you just open up a new sheet, paste in the values, and then save it. And what that does is it makes sure that you strip out any kind of like hidden stuff from the bottom. It's just a little tip. And so at this point, I would save that and it would be good to go. Now, this would not be good because I just faked a bunch of data, doubled some up. I can show you that. Since I doubled it up, there's going to be lots of little duplicated values now, but that's because I faked it. So that's the idea. That's how you can use the GDC. Let me know if you have any questions, and good luck.